I'd first like to thank Professor Kim and the other organizers of the KSSCR for inviting me to give this talk and for a chance for my first visit to Korea. It's really kind of exciting to be able to come and learn more about your country here. And also thank Ki-suk Park and Young-suk Son for chairing and sponsoring my visit here. So the main reason for showing this first slide, it's older work that was actually done by Ki-suk Park when she was a postdoc in my lab. And I'm not going to talk about this today. She can tell you more about it. But I think it's a beautiful picture that I like to show in the first slide. So I'm going to speak about contact inhibition and regulation of tissue size and cell proliferation in different contexts. And I'm going to start from the point of view of the role of cadherins, but really talk a little bit more about the signaling pathways involved in regulating tissue growth and regeneration. And the concept I'll start with is this concept of contact inhibition of growth. It's a well-known concept, especially in the cancer field, where cells, when they reach confluence and become very dense in culture, but presumably in tissues as well, at least in differentiated tissues, tend to stop proliferating. And when they're transformed as cancer cells, they don't obey the fact that they're neighbors and they start over-proliferating. Now, several years ago, we provided evidence that contact inhibition, at least in some cases, was mediated by cadherins and catenins, mediating cadherin contact between cells, and that was mediated by the so-called Hippo pathway. So the Hippo pathway, for those of you who don't know, it's called the Hippo pathway because it was first discovered, if we look in the bottom of the slide, in Drosophila as a pathway that regulated organ size, controlled organ size growth. So in this case, you see the eyes just outgrow. They're not tumors, but the eyes overgrow when there's mutations in this pathway. And all the genes in the Hippo pathway are found in mammals as well, and one sees if you mutate the pathway for example, in the liver, you get overgrowth of the liver, and eventually tumors can form. So the pathway involves a kinase cascade, the so-called hippokinase, MST in mammals, activates the LATS kinase, which phosphorylates YAP and keeps it out of the nucleus. So the important thing is that YAP, when it binds to a transcriptional cofactor, binds TEAD in the nucleus, stimulates proliferation, inhibits apoptosis, and promotes tissue growth. And the Hippo pathway, per se, inhibits YAP activity by phosphorylating it and keeping it out of the nucleus. So YAP in the nucleus is pro-growth, out of the nucleus is inhibiting growth. And cadherins tend to keep YAP out of the nucleus. And I think a nice example of contact inhibition in this pathway is shown on the right here. This is just a culture of MCF10A cells. It's a homogeneous cell culture with a little wound in the middle of it, and you can see around the edge. And this is just staining for YAP. And what one sees is that in the dense regions where the cells are surrounded in full contact, YAP is cytoplasmic. But at the edge where the cells have lost some of their contact, YAP accumulates in the nucleus. It's a very potent transcription factor and regulates a lot of pro-growth genes. So these cells at the edge really have a very different transcriptional profile than the cells in the dense regions and promote cell growth. So I think that has a lot of implications for thinking about the edges of tissues, edges of wounds, and, for example, leading edges of metastatic cancer cells, that cells can, because of this contact inhibition, have a very different transcriptional profile at the edge. So I'm really not going to talk about the cadherins that much now because we actually going to talk a little bit about other mechanisms that regulate the interaction between growth factor mitogen pathways and the Hippo pathway. So it turns out many different things regulate the Hippo pathway. I pointed out that cadherins, but also tight junctions and polarity complexes actually activate the Hippo pathway and inhibit proliferation. And I like to think about these as junctions are measuring epithelial integrity. So when the cells are in contact, they inhibit proliferation. There's a mechanotransduction pathway via integrins that actually does the opposite, inhibits the Hippo pathway and sends YAP to the nucleus and promotes cell proliferation. There's a lot of interest in the mechanotransduction pathway. And then secreted mitogens like receptor tyrosine kinase and GPCRs also inhibit the Hippo pathway and send YAP into the nucleus. I'm going to talk more about that. So they're and this is the basic result done by a former postdoc, Rune Fan, in the lab. And it's a very simple experiment. We took these MCF10A experiments. If you add epidermal growth factor for 10 minutes, you can see these contact-inhibited cells send YAP into the nucleus very quickly. And we can measure via transcriptional readout and chromatin IP assays that these effect of YAP is consequential. It's regulating target gene expression. And this seems to be important for EGF-dependent growth in these cells. If we knock down YAP with different siRNAs, then EGF fails to stimulate active proliferation in these cells. So this is an important component, and actually other groups have seen something similar as well. So just a little bit of an overview summary of what I think is going on is that we have cell contact inhibition activating the Hippo pathway, inhibiting growth, inhibiting nuclear YAP and proliferation, where growth factors, of course, stimulate proliferation through known pathways. But in addition to stimulating these known, like MAP kinase and other pathways, there's also turning off the inhibitory brake of the Hippo pathway. So we have this interplay between the mitogenic factors and the cell contact inhibition. And a way to think about that was, this is taken from a publication from another group, Kim et al. In 2009. What is contact inhibition? What's really the concept? And basically what they showed is that contact inhibition doesn't shut down growth absolutely. It just raises the threshold for growth factor activation. That is, at higher cell contacts, it takes a lot more EGF or other growth factors to stimulate proliferation. So you can get changes, for example, this is a very similar picture to what I showed you before at the edge. There can be a constant level of growth factors throughout, but the cells experiencing contact inhibition will be resistant to the effect of growth factors, whereas the cells at the edge are more sensitive. So this kind of interplay happens. So I'm going to talk a little bit more about the actual details of the pathways, how growth factors regulate YAP localization, the Hippo pathway. And there's been 3 different models published. The one I'll talk about a little bit is via PI3 kinase pathway, which has some interesting implications. Ken Irvine's group, using Drosophila, found that EGF affected the Hippo pathway through the RAS-MAP kinase pathway, and others have talked about the Rho-actin mechanotransduction pathway. So when we're confronted with this original observation, and my lab was not an EGF receptor lab with expertise, we had to sort of try and figure out what pathway regulated by EGF was involved. And the way we approached it was just to do a screen of very small molecules that affect different legs of the EGF signaling pathway. And the experiment's fairly simple. You take these serum-starved contact-inhibited cells and treat with EGF, and it goes into the nucleus or serum, and basically ask which inhibitors blocked the ability of EGF to send YAP into the nucleus. And basically, to summarize, what we found is inhibitors of the PI3 kinase PDK pathway inhibited the effect of EGF, but many other inhibitors did not, MAP kinase, RAS, and interestingly, neither did rapamycin or the AKT inhibitors affect the pathway, and that's an important point I'll come back to later. So we see PI3 kinase, because these AKT and rapamycin and TOR are downstream of the PI3 kinase pathway, but not involved in this regulation of YAP. And we showed that this does work through the Hippo pathway, because if you add these EGF sends YAP to the nucleus, but PI3 PDK inhibitors block that effect, But if you knock down LATs downstream, then the inhibitors no longer work. So they take effect through the LATs kinase, which phosphorylates YAP in the Hippo pathway. And we also see that the PDK inhibitors stimulate phosphorylation of YAP. So these two argue that the PI3 kinase PDK inhibitors are acting through the Hippo pathway. And we have some biochemical evidence for interaction with the pathway, that PDK interacts with the Hippo complex, and it depends on the protein called Salvador, and we've mapped a region of Salvador that seems to bind to PDK and recruit it to the Hippo pathway complex. So a model, and this is something we're working on, but our hypothetical model is that Salvador is a scaffolding protein that binds to LATs and MST as well as PDK, and that there's some activity of a protein X that's mediating the effect of PDK on LATs to affect phosphorylation and localization of YAP. So this is just another summary. We think, as I mentioned, AKT inhibitors and TOR inhibitors did not affect this pathway. So we think we've identified another branch of the PI3 kinase pathway that's probably important for tissue growth. And that's we're thinking about here. If you, this is a modified version of a slide I just downloaded from searching Google for PI3 kinase pathway. And this is what you typically see, is that the PI3 kinase pathway has many upstream activators, but works through PDK, through AKT and TOR mostly, and other AKT targets. But as I said, we don't think that either AKT or mTOR are involved in this regulation of the Hippo pathway. So we now think that the Hippo pathway, the PDK, PI3 kinase pathway, also has a branch where it regulates growth through regulation of the Hippo pathway. And I think that's going to be important to explore in many different contexts as a new branch of this pathway. If that's the case, then one might imagine that other things that regulate PI3 kinase are important. And what we showed here is experiments looking at PTEN. So PTEN counteracts the PI3 kinase pathway by dephosphorylating PIP3. And what we see is that, in fact, PTEN inhibitors send YAP into the nucleus because it's activating. By inhibiting PTEN, you're activating the PI3 kinase pathway. We also found that in addition to the receptor tyrosine kinases, the integrin-mediated regulation of PI3 kinase through focal adhesion kinase is also involved, and FAK inhibitors inhibit YAP nuclear localization in response to integrin binding. I'm going to, for sake of time, skip some of the next few slides and just summarize that. Okay. So we think EGF, G protein coupled receptors, integrins through the FAK, and SARC are regulating Hippo pathway, and these are all promoting growth by inhibiting the Hippo pathway and sending YAP into the nucleus. So in this context, we know that PI3 kinase, for example, is an important mediator of tumorigenesis, especially in breast cancer and other kinds of cancers. And we wondered whether, and most people focus again on AKT and mTOR, but whether the Hippo pathway plays a role. We don't have a final answer to this, but I'll show you some of our early results. And this is the one slide where I have something about stem cells in my talk. So we used a mammary stem cell transplantation technique to test the role of YAP in the Hippo pathway in mammary tumorigenesis. And what you can do is take mammary epithelial stem cells from a donor mice, transduce them in vitro, and transplant them back into a de-epithelialized mammary fat pad of a host mouse. And when you do that, you can reconstitute the entire mammary gland branching tree. And this was developed by Vestweber and Verb and Macaris group. And this is just to show you, actually, if you knock down YAP alone, YAP and TAZ is a homologue of YAP. There's no effect on normal mammary growth, regeneration in this case. In contrast, if you knock down beta-catenin, which is known to be involved in mammary growth, it blocks the development of this mammary pattern. But we wanted to ask whether this pathway was involved in tumorigenesis. And what we showed in this experiment is that overexpressing a constitutively active form of YAP causes the formation of mammary tumors in about half of all mice. So it turns out to be a fairly potent oncogene. All by itself can cause large mammary tumors to grow. And in this experiment, this is a model for mammary tumorigenesis, the PYMT model. And by knocking down YAP and TAZ, we reduce the time course of that. We didn't block tumors entirely, but slowed the formation of tumors in mice. So these data argue that the YAP and the Hippo pathway are involved in mammary tumorigenesis. We haven't connected it to the PI3 kinase pathway yet, but that's next on the agenda. So this is a slide. I just want to highlight other papers in the literature that have that I think our data have some explanations for. So for example, there are several papers that have showed that there are AKT-independent mechanisms of PI3 kinase signaling in cancer, and people have explored those mechanisms. What I want to do is hypothesize that the Hippo pathway and YAP are possibly involved. Another interesting model of that is that KRAS-driven pancreatic cancer is mediated by PI3 kinase, and another paper showed that it was also mediated by YAP and the Hippo pathway. So our hypothesis is this PI3 kinase of YAP is the mediator of both these phenomena. And then from the point of view of stem cells, the point I want to, it's not everything showing up well here, but there's evidence that PI3 kinase induces part of the way, these are activating mutations in PI3 kinase that induce mammary tumorigenesis. And some very elegant studies showed, lineage tracing studies showed that they work by actually inducing multipotency, so that all the different expression of this oncogene actually causes all the different cell types of the mammary gland to develop. And what I was going to show you here that's not showing up on the slide is this beautiful data showing that YAP is important for maintaining multipotency in mammary stem cells. So again, we're hypothesizing that this PI3 kinase regulation of YAP is responsible for that phenomenon. And they remain to be tested. So in summary, we're proposing that the Hippo pathway is a novel new branch of the PI3 kinase pathway important for probably development and tumorigenesis. So I'd like to thank the people who did the work. This work was, a large fraction of it was started and done at the University of Virginia, where I was there up until 2 years ago. Run Phan did the work on the PI3 kinase and the Hippo pathway. Nam Kim has done all the work on adherent contact inhibition and regulation of the Hippo pathway and other things I didn't talk about yet. And then Hong Bin Lee did the work on the mammary tumorigenesis. And as I mentioned before, Ki Suk Park did that beautiful cover slide, but it wasn't on this particular project. She did some beautiful work on the epithelial mesenchymal transition in the neural crest that I didn't have time to talk about. And I moved my laboratory to Seattle Children's Research Institute about 2 years ago. And this is a picture of the building where we are. There's a new building coming up right next door. And again, Nam Kim has been a major person pushing this project, and Alicia Mendoza is a new postdoc in the laboratory working on it. And here's a classic picture that everybody likes of Seattle with the Space Needle. The lab is right downtown here, and Mount Rainier in the background. So I hope some of you can come visit sometime in the future. Thank you. Okay.